Hi, welcome to part 2 of cytology, introduction to cytology. And here we're going to focus on the cell genetics, that is just DNA replication and protein synthesis and other things, to discuss cell division and growth and cell specialization. Nucleus genetic material, that is DNA, exists in a form of chromatin material in the cell nucleus, as we discussed. Chromatin material is made up of the DNA and histone proteins. Types of chromatin materials, they can be euchromatin, which are active during transcription, heterochromatin, which are inactive. And in this structure, during the organization level of DNA, it appears as strands. There are two strands antiparallel to each other, as we'll be discussing later, the DNA. Then it goes into DNA helix, which is twisted strands that are then rotated or surrounds the protein, the nucleosome and the protein, to form the histone protein to form the nucleosome. The nucleosome will then condense to form the chromatin materials that are now able to fit inside the nucleus. These chromatin materials, during cell division, they will condense further to form the chromatids. And the chromatids are now two. Like when they cross each other, they become chromosomes, which are two sister chromatid. So it's from the DNA strand, then twists to form the DNA helix. Then when the DNA helix surrounds the histone proteins, they form the nucleosome. The nucleosome try to condense to fit inside the nucleus to form the chromatin. The chromatids condense further to form the chromosomes. Okay, which appears two sister chromatids. Chromosome contains 46 chromosomes that exist in 23 pairs in a human body. And a cell with 46 chromosomes is diploid. A cell with 23 chromosomes is haploid. That's mostly the germ cells. From first to the 22nd chromosomes, we call them autosomes, and then the third chromosomes are sex-linked chromosomes. And human karyotype, this is where we present the graphic representation of the chromosomes within the nucleus of an adult. Normal human chromosomes, we said, has 46 chromosomes, and they are written as 46XX or X46XY, depending on the karyotype of examined cell. So we can have an abnormalities where we have an extra pair of chromosome, either on 21st, we call it Down syndrome. We've seen Down syndrome kids, they have slightly bent fifth digits, they have white spots on the colored parts of the eye, flat round face shape, small nose, small mouth and ears. And we can also have an extra chromosome on the chromosome 18, and this we call it Edward syndrome, where mostly you have low set ears, club feet, overlapping fingers, heart and lungs. Sometimes the kids may have an abnormality on the heart and lungs. Okay. An extra sex chromosomes on a cell for the men, we can, if it's an extra, let's say 47XX or 47XY, we call it Klinefelter syndrome. Mostly it's characterized by the symptoms such as Klinefelter. You can have long arms and legs. We can have tendency to lose chest hairs, tall stature. Short stature is for the Turner syndrome, where we have missing chromosome. It can be X0 or 45XY as representation, numeric representation. So for the Turner syndrome, the patient mostly presents as short, peripheral. They have trick ovaries. There is low posterior hair neck and webbed neck, so forth, so forth. So Klinefelter we have an extra, but for Turner syndrome it's missing. But the numeric representation for it can be 47XXY, this will be a boy. 47XXX, this will be a girl, still from our karyotypic presentation and gender identification. So the Turner syndrome, there is only one sex chromosome, and the representation will be now, for the Klinefelter will represent 47XXY, but for the Turner syndrome will be 45 because we have a missing chromosome. So it will be 45X0. For the missing one, we don't put 45X, we just put 45X then 0 to show that it's not like an error. It's something that it's a missing chromosome. We didn't forget to add an X. 45 is X, it's 45Y0 to mean there's a missing X. We just didn't forget to add that X, yeah. Can have an extra chromosome in chromosome number 13. We call it Patau syndrome, where it affects where the child can present with cleft lip palate, microcephaly, or a polydactyl. Okay, that one was about chromosome. So the structure of a DNA, we saw it's arranged from the strand. We go to the helix, which is twisted strand. Then we go to the nucleosome, which is when the DNA is surrounding the histone proteins, then chromatids, and so forth. So here, the DNA structure is arranged in such a way that the outer layer and the inner layer forms a kind of a bridge. Let me say like a bridge. So outside we have sugar phosphates backbone, sugar and phosphate backbone. Then inside, like kind of at their tail, now we have the alternating base pairs, where we have the guanine nitrogenous bases such as guanine, cytosine, thymine, and adenine. So they are arranged in an alternating way where we have a nitrogenous phosphate. Then we have for an sugar molecule. As we can see in this image here, there's pairing alternating, like an adenine is pairing with a thymine, cytosine is paired with guanine, like that. Then here in their bases we will have phosphate alternating with a sugar molecule on the base pair, like that. So the sugar is the deoxyribose sugar. So the red part is the phosphate, attaches to the deoxyribose sugar, then deoxyribose sugar, phosphate, deoxyribose sugar, phosphate, deoxyribose sugar, like that, up to the top part. The double-stranded molecule allows each strand to complement each other. The nucleotides are made of phosphate group, deoxyribose sugar, and nitrogenous base, as we discussed earlier. So they are they complement each other. Like for example, in this image, as we said there on the last slide, nitrogenous base are in the middle. Then the phosphate, the base, the backbone, is made of the sugar, which is attached to the phosphate, the deoxyribose sugar. So the nucleotide composed of phosphate, sugar, and the bases, nitrogenous bases, which arrange, and they form now the DNA structure. As we said, its backbone is made up of the alternating phosphate group and deoxyribose sugars. Nitrogenous base form the teeth that attach on the chain of the complementary strands. So each base has a pair, guanine, cytosine, thymine, adenine. The DNA is also antiparallel, where I describe the orientation of the two complementary strands that run parallel to each other but in opposite direction, from the carbon number 5 to carbon number 3, the attachment of nitrogenous bases and the phosphate. So the phosphate here attaches to the prime 3, that is the carbon number 3, but on top it will attach to the 5th prime. Oppositely, it will attach to the 3rd prime up to the 5th prime. So that is the antiparallel nature of the DNA. We can discuss more about the features of antiparallel DNA diagram. So directional ends. Each DNA strand has a distinct 5 prime end and 3rd prime end, where 5 prime end marked by phosphate group attached to the 5th carbon of the deoxyribose sugar. Then the 3rd prime end is marked by hydroxyl group attached to the 3rd carbon of the deoxyribose sugar. Opposite orientation. In the double helix, one strand is oriented 5 to 3 primes from top to bottom, while the other is oriented 3 to 5 from top to bottom. There is also a complementary base pairing, where the two strands held together by hydrogen bonds between specific pairs of bases, where adenine pairs with thymine, guanine pairs with the cytosine. Also, there is a feature of sugar phosphate carbon, where the outer side of the ladder, the backbone, are made of alternating sugar and phosphate groups, forming structural carbon of each strand. Hydrogen bonds, these bonds connect the nitrogenous bases in the center, forming the ranks of the ladder-like. Why do you think antiparallel DNA is important? Because during replication, enzymes like DNA polymerase can only synthesize new strand in the 5 to 3rd prime direction. So the antiparallel setups allows both strands to be copied simultaneously, though through different mechanisms. So you'll have the leading strand, which has 5 to 3, the normal required. Then you'll have the lagging strand. Also, the stability, the opposite direction, allows the optimal hydrogen bonding between the complementary bases, which contributes to the stability of the double helix. During transcription, antiparallel orientation is also crucial for transcription, where RNA polymerase reads the template attended a strand to synthesize messenger RNA. We will discuss this during genetics more, but that's just the intro part of it. So let's discuss the DNA replication. It's coordinated effort of multiple essential enzymes and proteins ensure that accurate timely duplication of a cell genome. So it occurs before mitosis. So it's just duplicating that cell has the same genetic information as the parent cell. And there are stages in DNA replication. Stage number 1, the two original DNA strands separate at the replication forks by DNA helix, helps in that. And the DNA polymerase now adds complementary nucleotides to each other as it goes like that in the leading strand. Now the more the RNA prime is there, then it copies as the strand goes ahead. Stage 3, the two DNA molecules formed that are identical to the original DNA molecules. So in a copyright proofread, the polymerase will still proofread if there is any mistakes that are done and so forth. So in this image, you are having the leading strand, which is, we said, it's from 5 to 3rd primer, which is the required one. So of course the DNA helicase will cut at the replication fork. The DNA polymerase will start duplicating. In the opposite lagging strand, it will have to, DNA polymerase will have to start from 5 to 3rd, then cut off, then start, like they'll form some of fragments. We'll discuss more as we continue. Insertion stage is the first stage during DNA replication. We said helicase enzyme facilitates unwinding of the DNA strand. Then stage 2, elongation, where the DNA polymerase brings the DNA bases to complement the template strand, synthesizing a new strand base. The 3rd stage is termination, where the DNA replication is stopped When the original strands are bound to their own finished complementary strand, two are identical DNA molecules therefore formed. Proofreading will help prevent accidental addition of inappropriate nucleotide that might cause genetic dysfunction. This is done by the DNA polymerase. In summary of the essential proteins and enzymes during the DNA replication, we said helicase help in unwinding and separating the double-stranded DNA at replication forks, breaking the hydrogen bond between nitrogenous bases. DNA polymerase, a complex enzyme which will catalyze the addition of new nucleotides to grow DNA strand. So it adds nucleotides in the 5 to 3rd primer direction and has proofreading function to remove errors, as I said. But now this will occur in the leading strand with no disruption. In the lagging strand, we will see what will happen. So in an RNA polymerase, they synthesize short RNA primers on both the leading and the lagging strand. DNA polymerase needs this primer to begin adding the nucleotide. So before it starts adding the nucleotide, the primase, enzyme primase, RNA polymerase will act like a catalyst to start there. So when it adds on the end, the 5th end, it starts now the transcription. So this one is needed both in the leading and the lagging strand. Single strand binding protein, SSBs. These proteins bind to the separated DNA. After helicase has separated it, this protein will bind to the separated DNA strand, preventing them from repairing. Then it also protects them from degradation during replication. Then we have DNA ligase. This seals the gaps between the fragments. During lagging strand, it will form in fragments, like it will add from 5th to 3rd primer, then go to another genetic arrangement from 5th to 3rd. So for the ligase, it will cut off those fragments that were formed, which they are called Okazaki fragments, on the lagging strands, creating a phosphodiester bond, forming the continuous strand from 3rd to 5th. We have a topoisomerase, is an enzyme relieves the strain on the DNA ahead of the replication fork, which is caused by the maybe unwinding of the helix, and it works by creating temporary nicks in the DNA and then resealing them. In prokaryotic, we have a DNA gyrase that acts as a polymerase in performing this function. So that is DNA replication. And we'll see expression, gene expression. Gene, of course, it's a DNA-based sequence that codes for the type, order of amino acid. Each gene provides a code to construct a particular protein. The gene transforms the information coded to final gene products, and we can see this in the protein synthesis and so forth. So after the DNA replication, the DNA has to participate in protein synthesis, but it can't move from the nucleus. So we'll have to synthesize the protein, then it will be transported, some kind of process. So protein synthesis occurs in the cytoplasm, and we need the DNA information to help us in the protein synthesis. So that is where we need DNA to RNA transcription and so forth. So there are several processes that takes place during the protein synthesis. Stage 1 is transcription, carrying the DNA information from the nucleus to the cytoplasm, and this one will be done by the mRNA. Translation is copying that information exactly the same way it is. Codon and other amino acids will be also arranged. Post-translation and modification, packaging of the DNA, exocytosis, it's done now. Every stage is done. So here, after transcription, which is mRNA, translation, we'll need the ribosomes to do BC. Then the post-translation modification, we'll need the Golgi apparatus and also rough endoplasmic reticulum to help us here. Exocytosis will be done with the transport vesicles. So first one we said it's transcription. It's the formation of mRNA from gene sequence, occurs within the nucleus and released by cytoplasm. When ready, guided by the RNA polymerase, will have different stages. So characteristics of RNA is single-stranded, unlike the DNA, which is double. RNA, ribose sugar in RNA has an additional oxygen. That is good to know, important, has uracil instead of thymine. The stages is the same as the DNA replication: insertion, elongation, and termination. So during insertion process, it's triggered at the promoter. Elongation, the RNA polymerase then unwinds the DNA segment. The one strand, the coding strand, becomes the template with genes to be coded. So this one codes just like copies in a coding, like the first three, like this is amino acid, copies it first, then goes to the other one. So RNA polymerase then aligns the correct nucleic acid with complementary base on the coding strand of a DNA. RNA polymerase then adds new strands to a growing strand of mRNA. Like its elongation is growing, the process of growing. So the one strand, coding strand, becomes the template with the genes to be coded. Template is something to copy in as you draw, as you do that things. RNA polymerase then aligns the correct nucleic acid with complementary bases on the coding strand. We said if G, A is 4, G is 4, like that. So it aligns if it's amine and thymine, and we said thymine will be replaced by uracil in RNA, so it just copies like that as it arranges in the complementary strand. Then the last stage is termination, when the RNA polymerase reach the end of the gene, one of the three specific triplets, either UGA, that's uracil, guanine, and adenine, or uracil, adenine, adenine, or uracil, adenine, guanine. Either of these three triplets will code a stop signal that triggers the enzymes to stop transcribing, and then the process, the mRNA will be ready, then it will be moved to the cytoplasm. So post transcribing, there will be a pre-mRNA undergoes a process called splicing. Spliceosome protein structures attaches on the mRNA and cuts the non-coding to remove non-coding regions of pmRNA. We'll have some areas that are not coded very well, or there were maybe mistakes, so it will have to remove it. So this removed part is intron and the remaining part is axon. Then the axon, now it will be the best, maybe the proofread that is the correct one, then it will be moved to the cytoplasm for protein synthesis. After transcription, we say we translate that information. So it's the formation of polypeptide chain from an RNA codes. It normally occurs within the ribosome. It involves reading the mRNA 3 bases at a time. If it's arranged like UGAA, it will have to copy it at that time to proofread it right, then go to another 3 bases, triplets, something like that. So still the stages are the same, 3 stages: insertion, elongation, and termination. So during insertion, binding of the ribosome on mRNA transcript will occur. Then elongation stage involves recognition of transport RNA, tRNA anticodon with the next mRNA codon in the sequence. tRNA then presents its amino acid cargo, and growing polypeptide strand is attached to the next amino acid by the help of an enzyme. tRNA releases the mRNA strand that shifts one codon over in the ribosome, and the appropriate tRNA arrives with its matching anticodon. Then, at the end of the process, final codon provides a stop signal. The final one here, now we don't have like restrictions, the final codon will provide a stop signal which terminates the translation process. This will trigger the release of newly synthesized protein. So after that, of course, after we've done the transcription, we transport it to the rough endoplasmic reticulum. If the cell, it's those proteins we needed to transport to the membrane or to the outside the cell, the ribosomes will release them into the rough endoplasmic reticulum. The rough endoplasmic reticulum will transport it to the Golgi apparatus that will package and sort it and transport it to exocytosis and other things. Yes, that was protein synthesis and DNA replication. Cell growth and division. So somatic cells divide regularly, that is except for germ cells. The following cell doesn't undergo cell division: red blood cells, gametes, neuron, and some muscle cells, like cardiac muscle cells. Its cell cycle is the sequence of events in life of a cell from moment it's created at the end previous cycle of a cell division until it's then, today or an hour ago. Divides itself, generating two new cells, that is mother and daughter cell. It has two phases, interphase and mitotic phase. So the interphase involves growing phase after cell division, synthesizing phase of DNA, that is DNA replication, protein synthesis, and so forth. G2 checks, confirms, and also continues growing, then preparing the cell to cell division. So mitotic phase is the cell divides the DNA cytoplasm, forming two new daughter cells in different phases, that is prophase, metaphase, anaphase, telophase. We'll have a stage after meiosis before going to G1 called G0. It's a resting state where the cell performs its function and is not preparing for division. For example, like we said, neurons, they don't do cell division, so it will be taken to G0 phase. Or if there was a mistake during mitosis, or it's not yet done, they'll have to go to G0 first before they continue with the normal cycle. Okay. The cell of course grows. I said interphase involves S phase, which is synthesizing phase, G1, which is gap1 phase, and G2, which is gap2 phase. Cell continues to grow and makes necessary preparation for mitosis. So cell mitosis, we said it involves four phases: prophase, chromosomes condensed during the prophase phase and become visible. Spindle fibers here, they will emerge from the centrosome as we discussed in our introduction lecture. Then nuclear envelope will break. Centrosomes will move toward the opposite ends. Can see there is a microscope image of prophase. Okay. Prometaphase is the late stage of prophase. Chromosomes will continue to condense. Kinetochores appear at the centromeres. Mitotic spindle microtubules attach to the kinetochores there. So it will stretch and come to attach at the kinetochores. Metaphase, chromosomes will align at the metaphase plate. Each sister chromatid is attached to a spindle fiber originating from the opposite poles. Then the anaphase part is centrosome split in two. Sister chromatids will move to the opposite direction, containing certain spindle fibers. So the remaining spindle fibers were not attached to the sister chromatids. Will continue to elongate the cell or move the cell opposite phases. The telophase, chromosomes arrive at the opposite poles and begin to decondense. nuclear envelope will appear and start surrounding the new chromosomes. The mitotic spindle breaks down, then the spindle fibers, the ones that were not attached to the sister chromatids, will continue to push the poles apart. Then at that process will have a cytokinase, the splitting of the cell or cleavaging of fura of the cell into two daughter cells. During cell division, there is a cell cycle controls to prevent outgrowing or mutation of the cell. So it involves molecules with cells and external triggers which provide stop and go signals. Each phase of division involves a process that must be completed before moving backward and is controlled by checkpoint. At checkpoint, the cell can be signaled to go or stop. Cyclin is a primary class of molecule that helps with checkpoint. Cyclin-dependent kinase work with cyclin to check progress point of a cell division. Cell differentiation is a process in which unspecialized cells become specialized to carry out distinct functions. Can be totipotency, pluripotency, multipotency, or oligopotency. Stem cells are present at different stages of life, like embryonic cell. Stem cell have an embryo which can do a lot of differentiation. Stem cell, fetal stem cell have fetus, and adult stem cell have an adult. We have different cells like chondroblasts in a bone cell, which are at an adult stage. We have some cells that did the division at an embryo and there's no more like any specialization can occur. We have some cells that proceeded to fetal stage, that mesenchymal cells that differentiated more into osteogenic cell and osteoblasts. The osteoblasts are now adult stem cells that keep on differentiating into osteocytes and other things.